Hey, welcome. So today we are diving into this really mind-blowing framework from a researcher named Zachary David Enfield. He's proposing that one single universal material could completely reshape our world. And get this, it's derived from one of nature's most misunderstood creations. I mean, just think about that for a second. What if we didn't need all these different metals, plastics, and composites? What if there was just one single, programmable substance, a single material that could be the foundation for everything we build, from our phones to our skyscrapers? And let's really unpack what that means. We're talking about a beam in a skyscraper that isn't just holding the building up. No, it's also acting as a computer chip, processing data, and it's a power source, generating its own electricity. I know, it sounds like pure science fiction, right? Well, according to this framework, it's a lot closer to reality than you might think. Okay, so here's the twist. The secret blueprint for this stuff, it doesn't come from some exotic element mined from an asteroid or a super complex lab experiment. Nope, it's based on the logic of prions. Yeah, you heard that right. The same prions we usually associate with some pretty nasty diseases. But the idea here is to completely flip the script and harness their unique properties as a revolutionary building tool. So how on earth does that actually work? Well, to really get it, we need to zoom in and look at the fundamental architecture that makes this whole thing possible. All right, at the very heart of this idea is something called cross-beta architecture. Basically, it's a very specific way that proteins can fold themselves and then stack together to create these incredibly stable, self-assembling fibers. The best way to think about it isn't like a chemical recipe. It's more like a universal blueprint for molecular origami. And this is the absolute key to the whole concept. The real power isn't in some special magical super protein. No, it's all about the fold itself. It's the way it's structured. And because the structural logic can be applied to almost any protein, it means we can engineer a nearly infinite number of variations of this material. We can tune it for basically any purpose we can dream of. Now, this is a really important distinction to make. Strength versus toughness. They sound similar, but they're not. Think about it. Steel is super strong, right? It's really hard to bend. But this amyloid architecture, it's incredibly tough. That means it can absorb a massive amount of energy before it actually breaks. And it does this while being extremely lightweight. So yeah, it's much more like spider silk than a steel girder. And its resilience? I mean, it is completely off the charts. It's resistant to the very enzymes that chew up normal proteins. It's stable in crazy high temperatures. It's hardened against radiation, which, makes it perfect for applications in space. And it basically just laughs at harsh chemicals. We're talking about one of the most durable structures science has ever seen. So, okay, we've established it's an unbelievably tough and resilient building block. But it's so much more than just that. A better way to think about it is as nature's smartest brick. See, it's this multifunctionality that really makes it a game changer. It's not just a passive brick you use to build a wall. It's a structural material that is also a conductor, also a sensor, and also an energy harvester. And here's the most critical part. It's self-replicating. It literally builds itself. And each one of these built-in properties just unlocks a whole universe of new applications. That mechanical toughness, that gives you self-repairing materials. Its natural conductivity, that lets you build self-assembling biological circuits. That weird property, piezoelectricity, it means it can literally create power from vibrations. And the whole self-assembly thing, well, that just completely rewrites the rules of manufacturing. All right, so let's get to the fun part. What could we actually build with a material like this? Because honestly, the possibilities are just staggering. Okay, first up, the obvious one, next-generation materials for building stuff. But yeah, we are not talking about your standard carbon fiber here. So check out this number, 160 megajoules per cubic meter. That's a measure of its toughness, how much energy it can soak up before it fails. And to put that in perspective, natural spider silk has always been the gold standard for biomaterials. Well, these engineered amyloid fibers don't just match it, they blow right past it. And that incredible toughness leads to concepts that sound straight out of a movie, like a self-repairing hull. Just imagine a spaceship, way out in deep space, gets hit by a tiny meteoroid. The damage exposes the ends of these fibers, and those ends then act like seeds, automatically triggering new growth to fill in the crack. The ship literally heals itself. But remember, it's not just about structure. This material can power and control itself. It's an active material, not just a passive chunk of stuff. And a big reason for that is a property called piezoelectricity. All that means is that it can generate an electric current when it's squeezed or vibrated, from mechanical stress, like vibrations in a building, or even just sound waves. And as you can see from this chart, these amyloid fibrils are way, way more effective at doing this than a lot of the conventional materials we use for it today. Okay, a material that builds itself is amazing, but it brings up a really big question, right? How do you control it? How do you tell it what to build instead of just ending up with a big blob of stuff? The answer is, well, it's as elegant as it is revolutionary. You use light. The whole process is called Prion optogenetic lithography. It works like this. First, you engineer these proteins so they're inactive. They won't do anything until they're zapped with a specific color of light. Then you just shine a patterned light, like a 3D hologram, into a vat of these proteins. And then the structure just grows, but only where the light hits it. It builds a precise 3D architecture, literally following the light as a blueprint. I mean, this just turns manufacturing completely on its head. Instead of taking a big block of something and carving away what we don't want, that's top down, we start with nothing and grow what we need, molecule by molecule. That's bottom up. You could grow a circuit board or a medical scaffold for your body or even a food lattice, all of it assembled with perfect precision, all programmed by light. So when you put all these pieces together, a self-assembling, multifunctional material that you can literally program with a flashlight, you start to realize we're not just talking about a new product. We're looking at the blueprint for a genuine leap forward for our entire civilization. The framework even lays out a potential roadmap. It starts pretty soon, between 2025 and 2030, with industrial-scale production and things like biosensors. Then from there, into the 2030s, we're talking aerospace panels in hybrid circuits. By the 2040s and 50s, we could see those self-repairing hulls on deep space vehicles. And then beyond 2060, well, that's when we see full civilizational integration. And that's really the ultimate vision here, the end game. A fully self-replicating, self-repairing material platform that works on a planetary scale. An infrastructure for our entire civilization that is truly sustainable. One that can grow, heal itself, and adapt, all built from the most basic biological ingredients. I think we'll end on this powerful quote from the author of the framework, Zachary David Enfield. It really gets to the heart of it. The question is not whether prion/amyloid architecture will transform civilization. The question is whether the civilization that does the transforming will understand what it is holding. And that is a heck of a thought to leave you with. Really makes you think.
I want you to imagine a material for a second. Just, try to picture a substance that is physically tougher to break than a strand of spider silk.
Right, which is already incredibly tough.
Exactly. But at the same time, picture this exact same material conducting electricity. Like functioning essentially like a microscopic quantum wire.
Yeah, that's where it starts to get weird.
It gets weirder. Now let's add a layer of complexity to it. If you take a hammer to it, like if you fracture or crack it, it doesn't just stay broken. It spontaneously heals. It actually knits its own structure back together.
Which sounds completely impossible.
Right. And just for a final thought experiment here, imagine that this exact same substance can be printed using nothing but patterned ultraviolet light. And you can print it into a perfectly textured, nutritionally complete steak.
I mean, it really sounds like a paradox, doesn't it? You're basically describing the properties of heavy industrial steel, advanced quantum electronics, self-healing living tissue, and, agricultural food products. And you're suggesting they all overlap in one single substrate.
Yeah, on the surface, it sounds like we're pitching some magic material from a sci-fi novel set 1,000 years in the future. But we aren't looking at science fiction today. Okay, let's unpack this. We are looking at a highly technical, rigorously documented 2026 manifesto.
Yeah, by Zachary David Enfield.
Right, Zachary David Enfield. And it's titled Prion and Amyloid Architectures, a Total Application Framework.
It is an incredibly dense piece of work, honestly. Because what Enfield has done here is he's aggregated decades of peer-reviewed experimental literature, covering everything from molecular biology to quantum physics to material science.
And he synthesizes it into this massive civilizational design framework.
Exactly. What's fascinating here is that scientists have spent basically the last 50 years looking at this material as a biological error. But engineering, and specifically Enfield's framework, sees it as the ultimate programmable matter.
And our mission for you, the listener, as we go into this deep dive today, is to completely rebuild your understanding of what this material actually is. Because the substance at the core of this framework, prions and amyloid fibers, is basically infamous.
Right, it has a terrible reputation.
Yeah, if you know the word prion, you probably know it as the terrifying agent behind these devastating neurodegenerative diseases like mad cow disease or Creutzfeldt-Jakob disease.
And that historical context is so crucial because medical science has spent half a century looking at the prion mechanism through a very specific, pathological lens.
You viewed it purely as a biological mistake.
Exactly, a deadly mistake in protein folding, where proteins aggregate into these indestructible plaques in the brain. But when you abstract away the pathology, when you look purely at the physics, the atomic geometry, and the mechanical properties of what this protein is actually doing, well, engineering sees something entirely different.
It makes me think of, the discovery of fire. Imagine if for the first million years of human history, we only ever saw fire when it was burning down our forests.
We would categorize it entirely as a forest-destroying hazard.
Right. It would take
Yeah.
a massive leap of imagination to realize, wait, if we control this, if we contain it, we can build a combustion engine. Or we can use it to cook our food.
That's a really great analogy.
So before we jump into how we might build a computer or a spacecraft out of proteins, we need to understand the structural foundation. Why don't these biological fibers just melt or fall apart the second they're exposed to the elements?
Well, that brings us to the core structural motif. It's known as the cross-beta architecture.
Cross-beta architecture, okay.
Yeah, and it's vital to distinguish here that we aren't talking about one specific genetic sequence of a protein. We are talking about a physical shape, a way that amino acids fold and lock together.
Because at the nanoscale, proteins are essentially just long strings of amino acids, right?
Exactly. And in a cross-beta structure, these strings fold back and forth to form flat planes called beta sheets.
So if I'm picturing this, it's not like a tangled ball of yarn. It's more like taking a piece of paper and folding it into a tight accordion fan.
That is a perfect starting visualization. Now imagine taking hundreds of those accordion fans and stacking them on top of each other. In the amyloid fiber, these beta strands orient themselves perpendicular to the long axis of the fiber.
Okay, so they're stacked incredibly tight. What's holding them together so strongly?
What holds them together is a massively dense network of hydrogen bonds, which are the same forces that hold water molecules together. But here, because the folding is so mathematically precise, the bonds create incredibly rigid, dense layers.
But rigidity alone isn't enough to make a supermaterial, is it? I mean, glass is rigid, but if I drop a glass window, it shatters instantly.
Right, it's brittle.
Yeah, so if this material is just tightly packed layers of rigid bonds, shouldn't it be incredibly brittle?
You're hitting on the exact problem that nature actually solved billions of years ago. Between these rigid hydrogen-bonded beta sheets, the protein has amino acid side chains sticking out.
Okay, side chains, what do they do?
Well, these side chains don't lock into rigid bonds. They interact more dynamically. They're flexible. They effectively act as molecular shock absorbers.
Oh, wow.
Yeah, so the nanocomposite structure of the fiber is alternating. You have a rigid layer, then a flexible shock-absorbing layer, then a rigid layer, and so on.
Okay, I think I see it. It's like nanoscale bulletproof glass.
How so?
Well, when a bullet hits bulletproof glass, it doesn't just hit a thick wall of standard glass. It hits alternating layers of hard glass and flexible polycarbonate plastic.
Ah, right.
The hard layer stops the penetration, but the flexible plastic layer stretches to dissipate the kinetic energy so the whole window doesn't just blow apart into dust.
That is a highly accurate analogy for the mechanics at play here. The kinetic energy from an impact travels through the amyloid fiber, but instead of snapping the main backbone, the energy is diffused into the bending and flexing of those side chains.
And we have actual data to back this up, right? This isn't just theoretical.
Oh, absolutely. The experimental data from atomic force microscopy and molecular dynamics simulations, which Enfield cites extensively up through 2024, proves this out. Scientists engineered a specific polymeric amyloid fiber using a short amino acid sequence designated FGASS.
FGAALSS.
Okay.
And what happened when they tested it?
When they tested it, they recorded a toughness of 160 megajoules per cubic meter.
Wait, let me stop you there, because 160 megajoules per cubic meter is a metric that most people, definitely myself included, just can't immediately visualize.
It's a very abstract number, I know.
Yeah, I know we throw around terms like strong and tough interchangeably in daily life, but in materials science, they mean very different things, don't they?
They do.
Mm.
And understanding the difference is key to why this material is so revolutionary. Think of Young's modulus as a measure of stiffness. Like, how much force does it take to stretch or bend a material?
Okay, so high tensile structural steel or carbon fiber would have a very high Young's modulus.
Exactly. They're incredibly stiff.
Yeah.
But if you apply enough sudden force to carbon fiber, it doesn't stretch, it snaps. It catastrophically fails.
Right.
Toughness, on the other hand, is the amount of total energy a material can absorb and permanently deform before it completely fractures.
So Young's modulus is a pane of glass. Very hard to bend, but shatters when the limit is reached, while toughness is more like a thick rubber tire, taking a massive beating and warping before it finally tears.
You've got it. Now let's put that 160 megajoules per cubic meter into perspective. Spider dragline silk is considered the gold standard in biology for toughness.
And what does that score?
It maxes out around 150 to 160 megajoules per cubic meter.
Wow, so this matches spider silk.
Yes. And high-performance synthetic polymers, like the Kevlar 49 used in body armor, sit around 30 to 50 in bulk toughness.
Wait, really? Kevlar is only around 50.
Yeah. And structural steel has a massive Young's modulus. But its toughness, its ability to absorb a sudden, violent kinetic impact without snapping, is significantly lower than this engineered FGAL's amyloid sequence.
So for you listening, let's just synthesize what that actually means. We are discussing a material grown from biology that equals or beats spider silk, absorbs more kinetic energy before fracturing than Kevlar or industrial steel, and, if I'm reading the data tables right, it does all of this at roughly 1/8 the mass density of steel.
It is phenomenally lightweight. But, the mechanical trauma is only half the story here.
What's the other half?
The chemical indestructibility of these fibers is what truly separates them from standard biological proteins. Normal cellular proteins, the globular ones doing the daily work in your body, are delicate.
Right.
If your temperature gets too high or the pH of your blood changes, they denature, they unfold and melt.
Like dropping a raw egg into a hot frying pan. The heat permanently destroys the delicate protein structure, turning it from a clear liquid into a solid white mass.
Exactly. But because the cross-beta structure folds so tightly, hiding its vital hydrogen bonds deep inside an impenetrable core, it shields itself from the environment.
So it just ignores the heat.
Heat, chemicals, you name it. These functional prion fibrils are heavily protease resistant, meaning the standard biological enzymes designed to chop up proteins simply cannot find a grip to tear them apart.
That's incredible.
And it goes further than biology. Enfield's framework details experiments where specific amyloid sequences like GNQQNY were submerged in pure boiling formic acid, or concentrated sodium hydroxide.
Which are conditions that would violently dissolve almost any standard biological tissue into a useless chemical soup in seconds.
Yes. Yet the cross-beta architecture remains entirely intact. Furthermore, because it lacks the continuous rigid crystalline lattice found in silicon or metals, it survives extreme gamma-ray radiation without structural failure.
Which is going to be incredibly important when we talk about deep space applications later on. But I have a logistical question here.
Sure.
If this stuff is practically invincible, how do we shape it? I mean, a steel beam is strong, but if I want a steel wire or a steel sheet, I can melt it down and forge it.
Right.
If this protein is so tough, how do we engineer it to do different things, rather than just growing a random ultra-strong blob in a vat?
That control comes from a principle called hydrogen bond density, or HBD. You can think of HBD as the master tuning dial for the material.
A tuning dial? How does that work?
Well, synthetic biologists don't need a furnace to change the material's properties. They just change the genetic sequence of the amino acids. If you want something analogous to structural steel, something permanent, rigid, and load-bearing, you engineer the sequence to pack those hydrogen bonds as tightly as physically possible.
And if I want something softer, say a material that can easily break apart and reform?
You adjust the sequence to lower the hydrogen bond density. You introduce slightly larger, more awkward amino acids into the core, which forces the sheets to sit slightly further apart.
Ah, so they can't lock together quite as tightly.
Exactly. This creates material that is dynamic. It might be fragile enough to intentionally fragment when exposed to a specific sound frequency, creating thousands of new seed points that then grow into more fibers.
So within the exact same biological manufacturing paradigm, you can tune your output from an indestructible architectural support column all the way down to a soft, self-replicating hydrogel.
Yes, exactly.
So if we take a step back, think about what this implies for the physical infrastructure around you. The framing inside the walls of your house, the chassis of the vehicle you drive, the bridges in your city.
It changes everything.
Currently, all of that requires mining raw iron from the earth, transporting it across the globe, and burning immense amounts of carbon in a smelter at thousands of degrees to forge it.
And this framework suggests a future where those materials are grown in a vat of water at room temperature using sustainable protein feedstocks, like cyanobacteria that literally pull their own carbon out of the air.
It's a complete paradigm shift in the energy economics of heavy industry.
It really is.
Okay, so we've grown a beam that can hold up a skyscraper. We have the structural foundation. But a modern civilization isn't just static walls and load-bearing columns. We require energy grids. We require massive data streams and computational power.
Right, the nervous system of the city.
Yeah. So if we replace steel and concrete with room temperature protein, do we still have to rip up the walls to run copper wire and fiber optic cables?
You wouldn't need to, because the material itself can be engineered to act as the wire. And this is where the framework transitions from material science into advanced quantum electronics.
Quantum electronics in a protein.
Yes. A landmark study published by Ships and colleagues in 2021 proved that individual amyloid crystals could transport electrons intrinsically over micrometer distances.
Wait, let me make sure I'm following this. Biological proteins are generally insulators, right? We don't think of meat or wood or hair as being highly conductive to electricity.
No, normally they aren't.
So how is an amyloid fiber suddenly acting like a copper wire?
It's not acting like a copper wire in the classical sense. In a copper wire, you have a sea of electrons that flow freely across the metal when a voltage is applied.
Okay.
The amyloid fiber achieves conductivity through a radically different quantum mechanical mechanism centered around a specific amino acid called tyrosine.
Okay, tyrosine. Let's break that down. What is it about this specific molecule that allows electricity to flow?
Tyrosine contains an aromatic ring. If you visualize it, it looks like a microscopic hexagon made of carbon atoms. Now, above and below this carbon hexagon, there's a cloud of electrons known as pi orbitals.
Pi orbitals, got it.
Normally, in a loose protein, these tyrosine molecules are floating around far away from each other, so those electron clouds don't interact. But remember the cross-beta architecture we just discussed, the tightly packed accordion fan.
Right, the dense folding that gives it the extreme toughness.
Exactly. When you fold the protein into that dense cross-beta scaffold, those tyrosine hexagons end up stacked perfectly on top of each other like a roll of coins.
Whoa.
And the geometric spacing enforced by the amyloid structure places each ring precisely 3.5 to 4.5 angstroms apart.
And I'm guessing that specific distance is the magic number.
It is the exact distance required for those pi orbital electron clouds to overlap. They basically merge into a continuous quantum pathway down the center of the fiber.
So it makes a tunnel.
Yeah. When an electron is pulled out of one of these rings, it leaves behind a localized positive charge, which physicists call a hole.
I always struggle with the concept of the hole in physics. So it's not a physical empty space like a pothole in the street.
No, it's not a physical void.
It's just the absence of an electron. And
Right.
because electrons repel each other, that absence can kind of move.
Think of it like a sliding tile puzzle. You know the ones.
Oh, yeah, with the one empty square.
Exactly. When you slide a tile into the empty square, the tile moves one way, but the empty square is effectively moved in the opposite direction.
That makes so much sense.
In the tyrosine stack, because of the perfectly aligned overlap, that hole doesn't just slide slowly, it tunnels quantum mechanically from one ring to the next. The charge hops down the entire length of the fiber.
So the protein itself is the circuit. The biological atomic geometry naturally forms the pathway, meaning you don't need to insulate it in plastic or wrap it in silicon.
Precisely.
Okay, that handles the wiring. But a wire just carries a signal. A wire doesn't compute. How do we get from a conductive protein string to an actual functioning computer brain? I mean, specifically relating to what the manifesto calls thermodynamic prion amyloid DNA, architectural electron and photonic advanced quantum computing brain-computer interface. It's a mouthful, but how does it actually compute?
Right, to understand that, we have to look at how we currently define a computer. Traditional silicon computing is fundamentally binary. You have a transistor, and it acts as a gate.
The voltage is either high, representing a 1, or low, representing a 0.
Exactly. Everything your computer does is a cascading series of ones and zeros. Enfield's framework introduces a totally different logic architecture. He calls it conformational computing.
Conformational meaning the physical shape of the thing.
Exactly. A prion fiber doesn't process information as a voltage switch. It processes information as a shape switch. A prion protein can fold into multiple, distinct, physically stable shapes.
Okay, so more than just two states.
Yes. Let's say it has a shape A and a shape B. Because of the unique templating nature of prions, which is actually the mechanism that makes them infectious in biological diseases.
Right, the zombie protein effect.
Exactly. If a protein in shape B touches a protein in shape A, it physically forces the shape A protein to refold into shape B.
So it's not a current flowing through a static wire, it's like physical shape-shifting dominoes.
Yes, dominoes is a great way to picture it.
If I introduce a chemical or an electrical pulse to the first protein at the end of the fiber and force it into a new shape, it knocks over the next domino. It physically twists the neighboring protein, which twists the next one, propagating a physical wave of structural change down the line.
And unlike a standard electrical signal in a wire, which gets weaker the further it travels due to resistance, this conformational signal is self-amplifying. Every time a protein refolds, it releases a tiny bit of thermodynamic energy driving the next fold.
That's wild.
And because these proteins can theoretically hold more than just two shapes, you aren't restricted to binary logic. You have non-binary multi-state logic gates.
Okay, I have to push back here. We're talking about quantum electron tunneling and incredibly precise microscopic physical shape changes.
We are.
Whenever I read about quantum computers, like the ones being built by IBM or Google, they are housed in these massive, multi-million-dollar liquid helium refrigerators.
Oh yeah, they look like giant golden chandeliers.
Right. And they have to be kept at absolute zero. Because if the environment is even slightly warm, the thermal energy vibrates the atoms, creates noise, and destroys the delicate quantum state.
Decoherence.
Yeah. So how does a delicate quantum signal survive in a vat of liquid or a warm biological body without immediately falling apart?
That is the standard classical intuition of physics, and it's the exact hurdle that skeptics raised. But if we connect this to the bigger picture, the answer lies in a phenomenon called vibronic coherence.
Vibronic coherence.
Which is a subset of a broader principle called cooperative robustness. To understand this, we actually have to look away from prions for a moment and look at green sulfur bacteria.
Green sulfur bacteria.
Yeah. Biology solved the room temperature quantum coherence problem billions of years ago in photosynthesis, specifically in a structure called the Fenner-Matthews-Olson, or FMO, complex.
Oh, I read about this. This is the bacteria that lives deep underwater where almost no sunlight reaches. Yet it manages to capture the few photons that do reach it and transport them to its reaction center with almost 100% efficiency. Physicists were baffled by how it didn't lose the energy as heat along the way.
Precisely. They discovered that the FMO complex puts the photon's energy into a quantum superposition, allowing the energy to essentially test all possible pathways to the reaction center simultaneously and instantly choose the most efficient route.
And it does this in a warm, messy, incredibly noisy biological environment.
Yes.
How? Why doesn't the ambient heat destroy the superposition?
Because the protein scaffold holding the conductive molecules isn't just a static, dead structure. The protein itself vibrates.
Okay, it shakes.
It has collective microscopic physical vibrations called phonons. What researchers found is that in specific biological structures like the FMO complex, and theoretically in the rigid lattice of an amyloid fiber, these phonon vibrations actually couple with the electrons.
They sync up.
They sync up. The heat of the environment creates vibrations. But because the cross-beta architecture is so structurally ordered, those vibrations occur at specific resonant frequencies.
Oh, I see.
Instead of the thermal noise randomly knocking the electron off course, the synchronized vibrations actually shepherd the quantum signal, protecting its coherence.
So it's not fighting the heat. It's surfing it.
That is a brilliant way to put it. The ambient thermal energy doesn't destroy the computation. The system is thermodynamically favored to use that ambient heat to naturally settle into the lowest energy state, which correlates to the correct logical answer.
That is breathtaking. I mean, the implications of that for you, the listener, are just profound.
It really solves a massive issue in tech right now.
Yeah. The biggest limit on our computing power today isn't making transistors smaller, it's heat. Data centers consume gigawatts of electricity just to run massive air conditioning units so the servers don't melt. What this framework proposes is a future processor that doesn't need a cooling fan. It doesn't need liquid nitrogen. It might actually run on the ambient room temperature. The thermal noise of your living room is the battery powering the molecular logic gates.
It entirely sidesteps the thermal bottlenecks that are currently choking Moore's law and traditional silicon scaling. And they could use ambient terahertz radiation to drive the state changes as well.
So if I accept that we have a material tougher than steel, and I accept that it can compute and transmit power at room temperature using quantum electron hopping.
Which is a lot to accept, but the data is there.
Right. But we still have a massive glaring manufacturing problem. How do I actually make a computer or a structural beam out of this? Because earlier you said that prion folding is a cascading, self-amplifying reaction.
It is.
So if I have a vat of liquid protein and I drop a single folded seed into it, doesn't it just uncontrollably crystallize the entire vat until it runs out of raw material? I'd end up with a giant, super strong rock, not a delicately patterned microchip.
That's entirely correct.
And I can't pour it into a hot metal mold because the heat would denature the precursor proteins before they could fold.
You're highlighting the exact limitation that stalled this field for years.
Yeah.
Self-assembly is great, but uncontrolled self-assembly is useless for precision manufacturing.
Right. So how does Enfield solve it?
This brings us to the third major pillar of Enfield's manifesto, prion optogenetic lithography.
Okay, optogenetics. I know opto means light. We use optogenetics right now in neuroscience labs, right?
We do.
Scientists take genes from algae that react to light, insert them into the neurons of mice, and then use tiny fiber optic cables to shine light into the mouse's brain to literally turn specific neurons on and off.
That's the exact technology.
But how does that apply to manufacturing a solid object out of a vat of liquid?
The framework proposes synthesizing that light responsive biology with the prion cascade. There are specific light-sensitive protein domains, such as Cry2 or LOV2.
Cry2 and LOV2.
Yes. These proteins naturally change their physical shape when they are hit by very specific wavelengths of light, usually blue or ultraviolet. What synthetic biologists do is fuse these light-sensitive domains directly to the prion-forming proteins.
Oh, so they act like a biological padlock.
Exactly. When the vat is kept in the dark, the optogenetic domain holds the prion protein in a closed, harmless shape. The proteins remain completely dissolved in the water, floating around like invisible building blocks, unable to lock together.
They're dormant.
They are completely dormant. But the exact microsecond a pulse of ultraviolet light hits the solution.
The padlock snaps open. The light forces the optogenetic domain to bend, which exposes the prion domain and instantly triggers that self-assembling cascade we talked about.
Yes.
But, and this is the crucial part, the crystallization only happens in the exact microscopic location where the light beam intersected with the liquid.
Correct. Now, combine that biological padlock with the technology we currently use to make computer chips, photolithography.
Right, where we use light to etch patterns.
Exactly. Today, we use lasers and arrays containing millions of microscopic mirrors to project incredibly complex nanoscale patterns of light onto silicon wafers. If you project a three-dimensional holographic light pattern into your vat of dormant protein soup,
Here's where it gets really interesting. It's like a sci-fi replicator. It's like developing a photograph in a darkroom, but instead of the light just chemically changing the color of flat paper, the light is physically freezing liquid into solid matter in 3D space.
that is exactly what is happening.
Wherever the hologram's light is brightest, the protein instantly crystallizes and solidifies. You just turn off the laser, reach into the vat, and pull out a fully formed, completely solid object.
No molds, no heat, no cutting, no waste.
And you can alter the physical properties of the object dynamically as it prints, right? We talked about the hydrogen bond density earlier.
You absolutely can. If the light pulses rapidly, perhaps it creates tight, high hydrogen bond density structures. If the light dims, it creates looser, more flexible structures, all from the exact same raw liquid.
I think this is where the sheer reality of this framework hits hardest, particularly when the text moves into discussing daily food production. Because we aren't just printing computer chips or building materials. The manifesto explicitly talks about printing protein lattices for human consumption.
The application for nutrition is staggering. When we look at the agricultural meat industry, we are essentially looking at an incredibly inefficient, macro-scale biological factory.
I mean, we use vast amounts of land, thousands of gallons of water, and months or years of energy to grow an entire animal just to harvest a specific arrangement of muscle and fat tissue.
Exactly. But with optogenetic lithography, you bypass the animal entirely.
How does that work in practice?
Well, you take a liquid protein feedstock, which you could grow using algae or cyanobacteria that literally just need sunlight, water, and atmospheric carbon. And you use the light printer to arrange those proteins voxels by voxel.
A voxel being a three-dimensional pixel.
Right. Because the laser can dictate the geometry of the fiber formation at the micrometer scale, you can print structures that perfectly mimic the complex microarchitecture of animal tissue.
So you're basically giving the light a blueprint of a muscle.
Yes, you instruct the light to align the tough amyloid fibers into dense, parallel bundles to simulate muscle fibers, and surround them with looser, water-retaining hydrogel matrices to simulate fat.
So you pull it out of the vat, and it has the exact physical snap of a fresh shrimp when you bite into it. Or you run a different software file, and the light pattern creates the precise muscular chew of a medium-rare steak.
All cruelty-free, all derived from carbon captured directly from the air.
Imagine what this means for your daily life. You don't go to the grocery store to buy a cut of meat that was shipped halfway across the country in a refrigerated truck.
No, you missed it.
You wake up, download a software patch for your kitchen appliance, and that file alters the holographic light pattern to print a meal tailored with the exact nutritional profile, amino acid balance, and texture you need for dinner that night.
It represents the complete decoupling of human manufacturing and nutrition from traditional industrial supply chains and geographic limitations, which is exactly why the manifesto takes this technology out of the kitchen and applies it to the most geographically constrained environment imaginable.
It's outer space.
Outer space.
Right, because if we can print a steak out of light and biological protein, what happens when we turn that HBD dial all the way up to maximum structural strength and try to print a spaceship?
The primary bottleneck of all space exploration is mass. Escaping Earth's gravity well requires an exponential amount of chemical energy.
Yeah, launching a single pound of titanium or steel into orbit costs thousands of dollars.
Exactly. Launching enough heavy metallic infrastructure to build a deep space vehicle or a lunar colony is economically paralyzing.
So we are sending an organic, carbon-based lattice into a radiation environment that routinely shreds metallic alloys. I mean, deep space is flooded with cosmic rays, solar flares, and high-energy particles. Wouldn't the radiation just absolutely cook a biological hull?
It is a highly intuitive concern, but it brings us back to the chemical invincibility we outlined in the first section. We have to ask why radiation destroys traditional electronics and metallic spacecraft.
Okay, why does it?
It's because metals and silicon rely on rigid, continuous crystalline lattices. When a high-energy particle, like a cosmic gamma ray, strikes a silicon microchip, it shatters that crystal structure.
Oh, it actually breaks the physical structure.
Yes, it physically knocks atoms out of alignment, creating a cascading defect that ruins the circuitry.
Ah, I see. But the amyloid fiber isn't a continuous crystal in that way. It's a cross-beta architecture. It's built of individual folded sheets with flexible, shock-absorbing side chains in between.
Exactly. It lacks that brittle, overarching crystalline lattice. The high-energy radiation passes through the cross-beta scaffold without causing the catastrophic atomic displacement that ruins metals.
So it might absorb the energy, but it doesn't shatter.
Correct. But radiation isn't the only hazard in space. The framework highlights another critical advantage of the cross-beta architecture: self-repairing aerospace hulls.
This is vital, because the vacuum of space is filled with micrometeoroids, tiny grains of rock or debris traveling at 20,000 miles an hour. They constantly impact satellites and the International Space Station, creating microfractures.
And right now, if a metal panel gets cracked, it stays cracked until an astronaut goes outside to weld a patch over it.
Right, which is incredibly dangerous and expensive.
Enfield draws a direct biological analog here to E. coli biofilm curly fibers, which are naturally occurring functional amyloids. Let's imagine a structural panel of our spacecraft is formed from an engineered amyloid composite.
Okay.
A micrometeoroid impacts the hull. The kinetic energy fractures the amyloid fibers, creating a small crack.
But it doesn't just shatter like glass because of the toughness we talked about earlier. It absorbs the blow, but still takes localized damage.
Correct. Now, the impact physically snaps the fibers, which exposes the broken ends of the internal beta sheets. These broken ends are highly reactive. They are packed with exposed hydrogen bonding sites that are thermodynamically desperate to stabilize. And because the spacecraft panel isn't just a dead, dry piece of wood, it's a composite matrix that contains dormant, liquid protein monomer suspended within it, those freshly broken fiber ends immediately act as new nucleation seeds.
Oh, that is brilliant. The thermodynamic drive to complete the hydrogen bond physically pulls the free, dormant monomers out of the surrounding gel matrix. The fiber spontaneously regrows from the broken end, chemically knitting the crack shut from the inside out.
It naturally heals the wound, driven entirely by ambient thermodynamics. There is no external repair mechanism required.
That's just amazing.
But it doesn't stop at physical repairs. The framework also addresses the energy scarcity of deep space, introducing a fascinating application for Dyson swarms based on the piezoelectric properties of the cross-beta structure.
Piezoelectricity, that is the phenomenon where certain materials generate an electrical charge when they are physically bent or squeezed, right?
That's right.
It's the same technology used in the clicker of a gas grill. You press the button, a tiny hammer hits the piezoelectric crystal, and it generates a spark.
Precisely. Now imagine we want to harvest energy in a distant solar system. We don't launch thousands of tons of heavy solar panels from Earth.
We launch a node.
Yes, a node, a compact seed package, perhaps a dry powder of engineered cyanobacteria. We land that node on a carbonaceous asteroid.
We hydrate the bacteria, they consume the asteroid's raw carbon and nitrogen, and use the ambient starlight to photosynthesize the liquid protein monomer. Then, an automated laser system begins printing.
It prints an immense ultra-thin membrane of amyloid fibers, stretching for miles in the vacuum of space. A biological solar sail.
But it isn't just absorbing photons like a traditional solar panel. Space isn't entirely empty. There is the solar wind, a constant stream of charged particles, and the physical pressure of radiation emitted by the star.
Exactly. And as that solar wind hits the massive ultra-thin protein sail, the membrane flexes, it ripples.
And because the material is piezoelectric, every single microscopic vibration, every bend and flex caused by the solar wind generates an electrical voltage. It passively harvests the mechanical stress of the vacuum into usable electrical power.
A self-growing, self-repairing, self-powering Dyson swarm, birthed from a single seed of genetically engineered bacteria dropped onto a dead asteroid. It solves the mass limitation, the repair limitation, and the energy limitation of space expansion in one unified architectural framework.
Which naturally leads to the massive question of timeline, because everything we've discussed, from room temperature quantum computing brains to printing shrimp out of light to self-healing asteroid space sails, sounds like it belongs in the year 3000.
It does sound like the distant future.
But Enfield's framework maps out an aggressive near-term timeline based on experimental benchmarks that were already being validated in labs prior to 2024.
The manifesto maps the transition in 4 distinct escalating milestones stretching from 2040 to 2060.
Wait, 2040 to 2060? That's barely a few decades away.
Yeah, we are discussing fundamental civilizational changes occurring within the lifetimes of most people listening right now.
Let's walk through those. Milestone 1 hits in the 2040s. This is the integration of amyloid-based computing co-processing with traditional silicon.
Notice that it doesn't suggest throwing away silicon immediately. That would be economically impossible.
Right.
Instead, it targets the immediate bottleneck.
Heat.
In the 2040s, we will see biological thermodynamic processors deployed for incredibly specific, high-density applications where heat dissipation is the limiting factor.
So if you are a major tech company trying to train a massive artificial intelligence model, you are constrained by how much electricity you can pipe into your data center to run the cooling systems.
Absolutely.
Milestone 1 suggests they will drop an Amyloid quantum coprocessor onto a standard silicon motherboard. The silicon handles the standard binary routing, but the complex heat-generating AI logic is offloaded to the protein chip, which actually uses the ambient heat of the server rack to power its conformational calculations.
Exactly. It's a hybrid transition. But by milestone 2, moving into the 2050s, the technology moves beyond niche computing and scales into heavy physical infrastructure.
And this is the deployment of self-repairing amyloid composite hulls on deep space vehicles.
Yes. Which makes sense as the next step. Once you've proven the material can survive the manufacturing process and hold a complex computational shape, you scale the volume up.
You stop launching heavy aluminum modules to the moon and instead send the cyanobacteria vats to print the habitats on site.
And that leads directly into milestone 3, slated for the later 2050s to 2060. The establishment of closed-loop protein lattice food systems fully operational on lunar and Mars installations.
Logistically, this is the only way a Mars colony survives. You cannot ship frozen food from Earth to Mars indefinitely. The launch windows only open every 2 years, and the mass penalty is too high.
Not to mention the farming issues.
Yeah, you also can't easily grow a traditional farm on Mars because the soil is toxic with perchlorates, the sunlight is incredibly weak, and traditional plants struggle in artificial gravity environments.
But a closed-loop optogenetic printer solves this entirely. The cyanobacteria grow in completely sealed, radiation-shielded vats, requiring only artificial light and carbon exhaled by the astronauts.
And then the printer takes that unpalatable bioslurry and restructures the cross-beta scaffolding to mimic the exact cellular texture and nutritional density of Earth-grown meats or complex carbohydrates.
It guarantees survival without relying on an Earth-based supply chain. And finally, we hit milestone 4, rounding out the 2060 timeline, the widespread commercial deployment of conformational logic neuromorphic processors.
Neuromorphic, so functioning like an actual brain.
This is the inflection point where the hybrid era ends. Biologically grown, non-binary computers enter the everyday consumer space. This raises an important question, though. How does our economic system handle a shift of this magnitude?
Because for all of recorded history, power and wealth have been dictated by access to physical resources. Mines, oil fields, heavy industrial manufacturing plants, global shipping lanes.
But this framework envisions a reality where physical manufacturing relies on genetic seeds, ambient carbon in the atmosphere, and starlight. You don't need a billion-dollar semiconductor fabrication plant. You need a vat of water and a calibrated laser.
The economic transition is absolute. The fundamental building blocks of our physical environment pivot from inert, extracted, finite resources to programmable, dynamically growing, renewable architectures.
It's comparable to the leap from the fragile, overheating, massive glass vacuum tubes of the 1940s to the microscopic, cool silicon microchips of the 1970s.
Except, instead of just applying that revolutionary shrinking and efficiency to calculators and early computers, we are going to apply it to all physical matter, every tangible object in your life.
It is a testament to the sheer power of reframing our scientific understanding. By looking past the pathology of a biological disease, we uncovered the universal engineering material.
And as we wrap up this deep dive into Enfield's incredible framework, I want to leave you, our listener, with one final, deeply strange thought experiment to mull over.
It's a good one.
Because if we follow the logic of this structural architecture to its absolute inevitable conclusion, we arrive at a very weird intersection. If your laptop's processor, the load-bearing walls of your apartment, the self-healing chassis of your electric car, and the steak on your dinner plate are all built out of the exact same programmable protein scaffold, what happens to the concept of physical waste?
It's a fascinating implication of molecular recycling.
If it's all exactly the same fundamental amino acids, just dialed to different hydrogen bond densities and twisted into different shapes using patterned light, could you, theoretically, take a broken, outdated quantum computer from your desk, toss it into a vat in your kitchen, use an enzyme or a specific acoustic frequency to dissolve it back into its base liquid monomer,
Yes.
and then use your optogenetic light printer to turn the dissolved remains of that old computer into a perfectly nutritious, perfectly textured meal for your family? I mean, in an amyloid-based civilization, yesterday's microchip is quite literally tomorrow's lunch.
The boundaries between material, machine, and living organism don't just blur, they dissolve entirely.
And that is something truly profound to think about the next time you look at the plastic and metal devices surrounding you today. Thank you so much for joining us on this deep dive into the cross-beta architecture of tomorrow. We will catch you on the next one.